The toy model brain. From bioelectrical to artificial intelligence. The brain is the bioelectrical computing center that controls behavior in advanced animal life. The brain is composed of electrically active cells known as neurons. A neuron is a cell that transmits electrical impulses. Typically, a neuron has a cell body, one of more pathways emanating from the cell body, or near it, typically called dendrites, and an electrically conductive channel known as the axon. The dendrites are connected to the axons of other neurons. When the neuron fires, it sends an electrical signal down the axon which, in turn, may lead to the activation of another neuron. Neurons can connect to muscles, triggering the muscle, and to sensory input where the neuron is triggered by the input. Neurons evolved before the brain, evolving more than 600 million years ago. Among the oldest species of animals with neurons on Earth are jellyfish. Jellyfish first appeared around 500 million years ago. Instead of a centralized cluster of neurons, a jellyfish has separate neural nets. One set of neural nets controls the activation of the muscles allowing the jellyfish to swim. The other sets, the rhopalia, receive sensory input, and control behavior. As evolution advanced, neural nets turned into a more centralized nerve system. Insects evolved around 400 million years ago. Insects not only possess a network of sensory and motor neurons but also a centralized neural processing center, a brain. One such insect is the fruit fly, Drosophila melanogaster, dew-sipper blackgut. The fly's brain is less than a millimeter in size but already contains over 100,000 neurons. Fruit flies display a number of relatively complex behaviors such as courtship, group formation, and combat. They also have a sleep-wake cycle of sorts, reminiscent of higher animals. Fruit flies have been the focus of extensive research. Recently, the entire brain of the fly was mapped down to the level of individual neurons and their connections. One of the primary reasons for the interest in the fruit fly is that its neurons on a basic level follow the same model as the neurons of more advanced animals including humans. Neurons in the fruit fly's brain use a combination of electrical and chemical signals to fire. When the electrical signal travels down the axon of a neuron, it leads to the release of chemicals, neurotransmitters, from the synapses at the end of the axon. The neurotransmitters reside in vesicles. When the electrical pulse reaches the end of the synapse, it causes pores to open that allow calcium ions to flow into the synapse. This is referred to as calcium channel current. These calcium ions allow the vesicles to fuse to the membrane of the synapse, releasing the neurotransmitters. These neurotransmitters latch on to receptors on the dendrite of the postsynaptic neuron and change the configuration of the receptors, causing them to open. The open receptors allow ions to flow into or out of the postsynaptic neuron. If the transmitters cause a net flow of positive charge into the postsynaptic neuron, then the neuron is primed to fire. If enough positive charge flows in, then the neuron fires. Inhibitory neurotransmitters bind to receptors that cause a net flow of negative charge into the neuron instead of positive charge. Such neurotransmitters inhibit the tendency of the neuron to fire. The neurons of the fruit fly actually use a number of the same neurotransmitters active in human neurons. These include dopamine, GABA, glutamate, acetylcholine, and serotonin. Some neurotransmitters affect many neurons in a region of the brain. These neurotransmitters modulate the neuron's tendency to fire. For instance, in the fruit fly, dopamine makes neurons in regions of its brain generally more active, promoting arousal, waking from sleep. Other neurotransmitters, termed FAST, essentially communicate between the presynaptic and postsynaptic cells, as shown earlier. These neurotransmitters are a fundamental part of the computational network of the brain. This biological computational network is the basis for the neural networks of artificial intelligence. In an artificial neural network, neurons are represented by nodes. Each neuron, or node, takes a number of inputs that range in strength from 0 to 1. These inputs are weighted, and based on the sum of the weighted inputs, the neuron outputs a fuzzy value between 0 and 1. The fuzzy value is typically generated by a step function, such as It can be shown that the equation for z is the equation of a plane in the space of the x's, the inputs, with a normal perpendicular to the vector formed by the w's. If the inputs, the x's, are graphed, then the node effectively creates a fuzzy plane that separates the trigger values from the null values. Each additional node adds another plane. If these nodes are then fed into a final node, the result selects a fuzzy shape from the space of the input values. In general, a neural network with one hidden layer makes a fuzzy selection of a simple shape in the hyperdimensional space of the input values. More layers of nodes can be added to allow for more general shapes. The computational network of the animal brain as simulated with AI is ultimately a fuzzy map of patterns in hyperdimensional space to output nodes that, in turn, trigger behaviors or other programs. Machines running artificial neural networks have been able to replicate a number of the functions of the animal brain, including object recognition, environment mapping, independent navigation and basic problem solving. As in other fields, the study of natural models has proven instrumental in the advancement of technology.